hopefully you'll be able to talk a little bit after the session. But next we're going to welcome Hsuan-Lin Her from Taipei Medical University, who is giving our next proceedings presentation on a pan-genome-based machine learning approach for predicting antimicrobial resistance activities of E. Coli.
So good afternoon, everyone. I'm an undergraduate student from Taipei Medical University, and I'm really happy to be here to share with our research titled A Pan-Genome-Based Machine Learning Approach for Predicting Antimicrobial Resistance Activity of the E. coli strains. So antibiotic resistance is really a huge problem. As you can see on the right, there are more and more pathogens with carbapenemase, an enzyme that can degrade one of the most powerful antibiotics to date. However, we are not having new drugs approved by the FDA. As you can see on the figure, there are less and less drugs approved yearly. So we definitely need some ways to detect resistance so that we can treat our patients fast and accurately. However, traditional methods rely largely on culturing methods, which usually take 3 days till we get the final results. So if we can utilize the genetic information well, then being fast and accurate can be possible. So to further define our research question, we want to build a model that takes the genetic information as the input and tells us about whether the pathogen is resistant or susceptible to a certain types of antibiotics. So existing model generally falls into 2 categories. The first one is knowledge-based models, meaning that they find no resistant genes within the query genome and then use those as the feature to predict the resistant phenotype. And those no-resistant genes can be further built into a deburring graph and for convenience of mapping the sequence reads. And the other category of models are largely k-mer-based, which means that they fragment all the query genomes into k-mers and use the distribution frequency as the feature vector to predict antibiotic resistance. So those 2 categories of models generally are achieving good accuracy. However, there are some drawbacks. For knowledge-based models, if there are some novel mechanisms, they may miss it. And for k-mer-based models, it's rather difficult for them to backward interpret what the features mean. Therefore, we propose our pan-genome-based method, and I will explain in the next slide why this might be a choice. So before I dig into what pan-genome is, I want to first explain how a pathogen can get resistance. So they may get resistance from mutation or horizontal gene transfer. So horizontal gene transfer means that the pathogen has access to a pool of genes that doesn't necessarily constrain within a single species. The resistant gene can be from a compatible plasmid or it can be from a phage. And pan-genome is a notion that tells us about the genes in all strains in a species. So say if that 2 bacteria belongs to a single species, then the pan-genome of that species will include all genomes here. So to train our model, we'll need some data. So we download the data from the Patrick database. And the data composed of 2 parts. The first parts are the genome sequences. And there are 59 strains of E. coli that has resistant metadata. And for the resistant metadata, there can be further divided into 2 parts. The first one is the minimal inhibiting concentration, meaning that the lowest antibiotic concentration needed to inhibit the growth of the bacteria. And also the resistant phenotype, which is based on the minimal inhibiting concentration. So how does the minimal inhibiting concentration goes into the phenotype is based on some clinical criteria such as COSI or UCAST. And as you can see, every entry of the data are based on different criteria. So I will further explain how these criteria works. So I will take COSI 2000 as an example. So the criteria gives us a set of breakpoint, the R breakpoint, the resistant breakpoint, and the susceptible breakpoint. So any MIC has greater than the R breakpoint will be classified as resistance, and those lesser than the S breakpoint will be classified as susceptible. Anything falls in between will be classified as intermediate. However, those criteria, they change over years. So say we have a bacteria that has a minimal inhibin concentration of greater than 8. It will be resistant in year 2000. However, in year 2017, it will be either intermediate or resistant. And that will cause a huge trouble in model training. So therefore, we annotated all the resistant phenotype according to the COSI 2017, and all phenotypes that are unsure are removed from our training. And then we move on to construct a pan-genome using the program CD-HIT. So say we have 3 bacteria, the red one, the green one, and the orange one here. So after all the open feeding frames are predicted, CD-HIT will sort them along with their length. And anything with greater than 95% amino acid identity will be clustered into a pan-genome cluster. So the longest genome will form the first cluster, and the second one, because it doesn't have that high identity, will form the next cluster, and so does the third genome. And for the fourth genome, because it has a high identity, so it belongs to the second genome, and here comes all the gene clusters. So as you can observe that there are generally 2 kinds of clusters. The first one are the core clusters, meaning that all strains of the bacteria has at least 1 gene in that cluster. And the remaining are the accessory clusters. So here comes our pan-genome. As more and more genomes we use, the size of the pan-genome grows rapidly, and also the accessory genome. The core genome gets smaller, and it converges to 2,874 in the end. So as the more genome we use, the growth of the pan-genome size doesn't plateau. So this corresponds to a pan-genome, indicating that the E. coli has access to an infinite size of genes. So then we annotate our clusters. We extract all the representing genes, and then use cluster of orthologous group to indicate their general functions. So the cluster of orthologous group tells us about what a gene does. So for example, in the first gene is annotated as class J, meaning that it has something to do with the ribosome. And also we use the comprehensive antibiotic resistance database to detect resistant genes. So for example, the first gene is annotated as QNRB, so that means that the gene is already known to be contributing to quinolone resistance. And we observed that some card categories are predominant in core genome, and those are mostly housekeeping genes, including amino acid transport, nucleic acid transport metabolisms. And also some categories are predominant in accessory genome, and those are predominantly defense mechanisms such as DNA repair. And we also found out that only a really small portion of genes are known resistant genes, composing only 0.7% of all the clusters. And for all the known resistant genes, only 60% of them corresponds to accessory genome. And for the core genes, core resistant genes, they may consist of intrinsic resistance. And then we move on to make the predictions. So we tried 4 kinds of model: AdaBoost, naive Bayes, random forest, and SVM. And we tried 4 different sets of genes as a feature. So the pink one denotes that all clusters are included. And the green one denotes that all accessory genes. And the blue one corresponds to all known resistant genes. And the blue one is all accessory resistant genes. So as you can see, that the blue one, the card accessory genes, has the best performance, telling us that rather small portion of the pan-genome can predict the antibiotic resistance rather great. And because the performance is not good enough, so we move on to use genetic algorithm to do the feature selection. So the candidate features are the 68 accessory and the CAR genes. So here, say we have 5 genes, and they are randomly selected. So in the first row, only gene B and D are selected as a feature. And so the first row corresponds to 1 GA genome. So we'll have a population of GA genomes. And then we use SVM to validate how good they are. And the two best GA genome with the best area under curve will be selected, and then cross over, mutated, and have a next population of GA genomes. So after 30,000 iterations, we'll have our final feature sets. So here comes the results. So compared to the red one, the 68 CAR accessory genes, our GA selected clusters improve a lot. And we also compare our GA selected clusters to Scori, which is a program that scores all the pan-genome clusters according to a phenotype. And also the ones we've also dig out the known resistant genes that are predicted to be relevant to resistance by Scori, and our GA selected cluster still outperform them. And we also compare our GA selected genome to existing literature. So Tyson et al., they tell us, like, for each class of antibiotics, what exact genes are relevant to the resistance. So for example, for beta-lactam antibiotics, the gene TM oxide, CMY, and MC are relevant. So in the Tyson model will only incorporate these 4 genes for the prediction. As you can see, our genetic algorithm features outperforms the literature-based feature selections on 4 different kinds of antibiotics. So we wanted to know that the features selected by our GA genome, does that correspond to known drug mechanisms? So in this figure, the green rectangle denotes 1 GA genome for a particular drug. So in the rectangle, the dark blue ones means that that gene is selected for prediction. And different columns corresponds to the GA genome for different drugs. And the GA genome are clustered hierarchically to see if there's any clustering of known mechanisms. And it turned out that those mechanisms are not related to the clustering of GA genome. So we can see that many cephalosporin class of antibiotics doesn't cluster within a single clade. So what really happened here? We went back to see what features are selected for prediction, and we found out that the selected feature doesn't necessarily correspond to the known mechanisms. And so, for example, we have a gene called PMRE, which are already known to be contributing to polymyxin resistance. But that gene is selected for cephalosporin's prediction and also some antibacterial folate pathway. And what's interesting is that Scori also make the same prediction. So there are a lot of possibilities. There may be some new mechanism that we don't know. There may be some co-evolution existing in the pathogen's genome, and maybe there's just some noise within these feature selection progresses. So the limitation of our research is that we only include 59 E. coli genomes, and in the future we wish to extend to more genome and species so that we can better validate our results. So our conclusion is that we use a genetic algorithm to select a subset of gene clusters for phenotype prediction, and our prediction area under curve outperforms current gene-based methods. In the last few slides, I would like to thank my advisor, Dr. Wu Yuwei, for his help in every aspect of this research. And I also like to thank all the founders, my school, Taipei Medical University, the Ministry of Science and Technology, and also the Foundation for Advancement of Outstanding Scholarship. Thank you for your attention, and I will take questions.
So how are the 58 strains that you considered related to each other, and is there a tendency for strains which have greater overall similarity to share resistance characteristics?
Can you repeat it again?
So you did a couple of trees of your prediction measures. If you just build a phylogeny for the bacteria, are the different clades, do they share resistance characteristics in these particular 58 strains that you were looking at?
Yes. We found that several strains are resistant to a lot of bacteria, while some only exhibit intrinsic resistance.
Well, so what about too closely related strains? So presumably the 58 strains are not all radiating from the same root in their own tree, right? So if you have clades in the bacteria, do those clades share? So the concern is that you're predicting which clade the bacteria are in, right? Thank you.
So maybe we will have to work further on this.